today is we're going to lay out the same kind of pattern we ended with yesterday. We're going to lay out that nephron, that particular kind of nephron called the juxtamedullary nephron. We're going to lay it out beginning with Bowman's capsule, the proximal convoluted tubule, and running it across into the depths of the medulla, and then back up to the cortex again. And so we'll run. But the next thing we really want to talk about, though, is the concept of the corticomedullary gradient. Now, the kidney functions by generating a gradient of ions and small molecules that ranges from about 300 milliosmoles, pretty much isotonic with plasma, in the cortex, deeper and deeper into the medulla, higher and higher and higher, until by the time we get to the depths of the medulla, we're up around 1,200 milliosmoles. 600 milliosmoles of that is from sodium chloride. The other 600 milliosmoles is from urea. And we'll see today, but certainly on Monday, how concentrating urine. So we can't do without the urea. But what we want to say is we want to lay out the nephron structures we did on yesterday. And then we're going to lay out the corticomedullary. Then we're going to think about the orientation and organization of the epithelial cells that make up the nephron. And we're going to start with the individual epithelial cells. Now, one of the hallmarks of this discussion is that as we move from one epithelial cell to the next along the length of the nephron, we change the composition of proteins in those cells. The proteins, the transmembrane proteins in the descending limb will be predominantly aquaporin. And the water channels to draw the water and into the interstitial space. Then we go around the hairpin turn and we start up the ascending limb. The ascending limb, by the time we begin talking about these cells have now lost their aquaporins. And they can't move water across their cytoplasm and into the extracellular space. And in this thin segment, we now begin to see that we can use simply a passive gradient to move ions out of the lumen of the tube and into the interstitial or extracellular space. But then there's a transition in the ascending limb from something known as the thin segment to the thick segment. The name gets its terminology because the cells get wider, they get thicker. And these thick segment cells have a different group of proteins, a different assortment of proteins. We'll see that within this thick segment we have these apical transporters, and we'll have basilar pumps. We'll see that among these epithelial cells in the thick segment that we have a sodium-potassium 2-chloride transporter on the apical side. This sodium-potassium 2-chloride transporter is going to be snagging ions in the lumen of the ascending limb, moving them into the cytoplasm, and then once in the cytoplasm, basilar pumps, mostly sodium-potassium ATPases, take over and they move those ions into the interstitial space. Now, for some distance along the thick segment, all we're doing is bringing the contents of the lumen of the ascending limb to equilibrium with what's in the interstitial space. But these pumps, these transporters rather, are so efficient that as the fluid moves higher and higher in the ascending limb, we can actually get the sodium and chloride levels so low that they're hypoosmotic. We'll actually get sodium and chloride levels in here of about 50 millimolar sodium and 50 millimolar chloride for a total osmolality of about 100 milliosmoles. So about a third of what you see in the cortex. And then finally, the ascending limb is going to empty into something known as the distal convoluted tubule. Now, for our discussion in here, the distal convoluted tubule is going to be the last station in the nephron where we can reabsorb sodium ions. It's a bit of a fib, but it works. It simplifies the story a little bit. And so once we get into the distal convoluted tubule and we have 50 millimolar sodium and 50 millimolar chloride on the outside, we can now simply use a gradient of the concentration of sodium inside the cell versus what's in the lumen. The luminal concentration of sodium is 50 millimolar. The cytoplasmic concentration of sodium in whatever cell you look at is about 10 millimolar. So now we have a 5-fold gradient. We can put sodium channels in the apical membranes. Sodium flows down its concentration gradient. So we lower the concentration of sodium in the lumen of the distal convoluted tubule, and then on the basolateral side we'll put pumps and pump that sodium out. What makes this region different from the rest of the regions we've talked about is that these proteins are under hormonal control. There's a hormone we talked about briefly before, we're going to spend some time on today, called aldosterone. Aldosterone. The sterone tells you it's a steroid hormone. When you hear the term steroid hormone, two things ought to come to mind. First, it's made from cholesterol. That means it's lipid soluble because it's whizzing right across the phospholipid bilayer, goes into the cell. But remember, the steroid hormones bind to cytoplasmic receptors. By binding to cytoplasmic receptors, then they're transported into the nucleus. And then that receptor hormone complex becomes a transcription factor, turns on particular genes. The effect of aldosterone in these cells of the distal convoluted tubule is to turn on the genes that make the protein's sodium channels, called ENaCs, epithelial sodium channels, and a set of other proteins in the basilar surface called sodium-potassium ATPases. So as the sodium comes in, the sodium-potassium ATPases are pumping those ions out the basolateral side. Then if there's time, and there wasn't in the first section, we'll begin thinking about something known as tubulo-glomerular feedback, and this is something that's going on in the distal convoluted tubule. You have to envision the idea that this fluid, which has started in Bowman's capsule, has now moved through the entire length of the loop of the nephron. And while in the loop of the nephron, we have various pumps and channels to remove the sodium and chloride. So now the sodium chloride becomes a kind of problem. Sodium chloride concentration tells us indirectly how long that fluid has been in the loop of the nephron. The longer the fluid has been in the loop of the nephron, the lower the sodium chloride concentration. The faster the fluid is moving through the loop of the nephron, the higher the sodium chloride concentration, simply reflecting the idea of how much time we've had to withdraw that sodium and chloride. So now the sodium and chloride becomes a kind of signal to cells in the distal convoluted tubule. These are the macula densa cells. Macula means dark spot. Macula densa means dark spot. And these are particular cells that play an important role in regulating glomerular filtration. And the process goes as follows. These macula densa cells are monitoring the concentration of sodium chloride in the extracellular space. They're monitoring the concentration of sodium chloride in the lumen of the distal convoluted tubule. So they're asking the question, how much sodium and chloride is out there in the lumen of the tube? That's reflective of how long the fluid has been in the loop of the nephron. So at that point, if sodium chloride is high, that means that the time that the sodium chloride is spent in the loop of the nephron is too short. It's moving through too fast. The flow is too high. And so the response of the macula densa cells to that high sodium chloride is to now release adenosine, probably in the form of ATP. That ATP is quickly converted to adenosine. There are now adenosine receptors on the smooth muscle of the afferent arteriole. And we'll see that all of this is strategically located right next to one another, so that the release of the adenosine can, in short distance, bind to the receptors on the smooth muscle. When the smooth muscle binds adenosine, it contracts. This is the smooth muscle of the afferent arteriole. So now let's walk through it again. Glomerular filtration is too fast. We're moving too much fluid from the blood vascular compartment into the renal compartment, and we know that because the sodium chloride concentrations are high. So the feedback system says, all right, sodium chloride concentrations are too high. What we'll do is we'll reduce glomerular filtration. We'll reduce the pressure, if you will, of the fluid in the glomerular capillary. So to reduce the pressure of the blood in the glomerular capillary. The adenosine binds to the smooth muscle and constricts it. That means less fluid can flow into the glomerular capillary. There's less pressure. That throttles back the glomerular filtration rate. That means that the fluid is now going to move more slowly through the loop of the nephron. Because the fluid is moving more slowly through the loop of the nephron, there'll be less sodium chloride arriving at the macula-densa cells. The lower sodium chloride concentration of the macula-densa cells to stop releasing adenosine. This is the constriction on the afferent arteriole, and now glomerular filtration rate can go back up again. So in this way, we're regulating glomerular filtration rate through the system. And then finally, there's going to be angiotensin, renin-angiotensin pathway. We'll probably get to this on Monday. It's affecting systemic blood pressure. And the way I tell the story here, and I think the way most people tell the story, is to talk about angiotensin pathway in terms of hypovolemic shock. And what we mean in terms of hypovolemic shock is that there's been tremendous blood loss. There's been an accident. There's been a knife wound or a gunshot wound, and people have lost blood. The response to that loss of blood is to increase the blood pressure. And the way we increase the blood pressure is we reduce the overall volume of the blood vascular compartment. And the way we reduce the overall volume of the blood vascular compartment is to cause that smooth muscle, which is running along all of the arteries and arterioles, but also, which we didn't talk about, along the venous side, and constrict them. So that effectively reduces the volume. So what happens is when there's reduced blood flow, that leads to the release of this enzyme called renin. There are special cells on the afferent arteriole that are monitoring the amount of stretch on the afferent arteriole, and when the amount of stretch goes down, because the volume of fluid in the blood vascular compartment has fallen, then what happens is renin is released. It's an enzyme that converts this plasma protein, angiotensinogen, from an inactive precursor form to something known as angiotensin 1. Then there's another enzyme that's all along the apical surfaces of endothelial cells called ACE2, this is ACE1, angiotensin converting enzyme. This is a relative, it's a protein relative of the ACE2, which is the receptor for the COVID spike protein. But this is ACE1, and ACE1 has a function of converting angiotensin 1 to angiotensin 2. Angiotensin 2 is a very powerful stimulant of smooth muscle contraction. So it will cause smooth muscle contraction throughout the cardiovascular system, and by doing that, increase this blood pressure by increasing the volume. And we'll walk our way through that. But let's start here with where we left off. Where this is one of the kidneys. And out here in the cortex, this is the renal cortex. And you'll remember we've emphasized the idea that all the blood flow coming into the kidney ends up flowing into the capillaries, the glomerular capillaries out here in the renal cortex. And we said then that in the renal corpuscle, where the glomerular capillary met Bowman's capsule, that was all cortical. So this is meant to be Bowman's capsule. This is meant to be the proximal convoluted tubule. And that then gave rise to a set of epithelial cells which formed this tube called the descending limb. Of the loop of the nephron. This goes down around the hairpin turn, and this forms the structure known as the ascending limb of the loop of the nephron. So the ascending limb then ends up here, emptying into this structure known as the distal convoluted tubule. This convoluted tubule is in the cortex. In the cortex, it empties into this structure, known as the collecting duct. I'll remind you that down here is going to be the renal down here is the renal calyx. And that the filtrate from the collecting duct is going to empty into the renal calyx, feed into the ureters that go into the bladder. But what we're interested in that we have idea I'm going to put forward here is the concept of the cortico-medullary gradient. Much of what we're going to talk about today revolves around the idea of generating this cortico-medullary gradient. But for now, let's just name it and give some values to it. When we look at the concentration of ions and small molecules out here in the cortex, we're in the neighborhood of about 300 milliosmoles. About 130, 140 of this milliosmoles is from sodium, another 110 from chloride, some from bicarbonate, some from some other ions. Now, that's the osmolarity. That's all of the ions. But as we walk from this cortical region and get deeper and deeper and deeper into the medulla, the concentrations can increase. And under extreme circumstances, the concentration down here in the medulla, right above the point where the collecting duct empties into the renal calyx, the concentrations down here can be in the neighborhood of 1,200 milliosmoles. These concentrations of about 1,200 milliosmoles are composed of 300 millimolar sodium and 300 millimolar chloride and about 600 millimolar urea. Remember what urea is. Urea is that waste product which has been generated from the catabolism of proteins. It's been generated from the ammonia, that's the waste product, and in the liver we generate urea. We're going to use this urea. We're going to use this high concentration of urea, which is a byproduct of protein degradation, and we're going to use that urea to get high concentrations, high osmolality in here that can come to equilibrium with the urine in the collecting duct. And that's how we're going to concentrate the urine. But that's a story for Monday. What we want to do today is we want to talk about how we can go from 300 millimoles to 1,200 millimoles. Now, I'm going to lay this out as a discontinuous gradient, meaning it's like a layer cake. We'll see higher and higher concentrations. In fact, it's a continuous gradient. And if we look at these layers, so I'm going to lay it out as a layer cake and put some numbers on it. But if we were to actually look at this in biology, we'd see that this is a continuous gradient. But it goes from 1,200 here to, pick a number. This is maybe, 900. This is maybe 750. This is maybe 600. This is maybe 450. This is maybe 350. Until we get out here into the medulla, where we're down to 300 milliosmoles. So the long and the short of that is that as we would swim from the cortex into the medulla, we see ever higher concentrations of salt, until down deep in the medulla, we get to 1,200 milliosmoles. We're going to use this extremely high concentration of urea and salts to concentrate urine in the collecting duct. But before we can use these high concentrations of ions and salts to concentrate the urine in the collecting duct, we have to generate the gradient. So what we're going to do now is we're going to go through all of the epithelial cells and talk about generating the gradient. And we're going to see that the nephron's major job, subset of the juxtamedullary nephron. Its job is to generate the gradient that can be exploited to concentrate urine. So let's take a look at a couple of pictures and see if we can kind of orient you with this image of the nephron. This first image is one that a TA a couple of years ago in the course drew. He's now in an anesthetist school in Milwaukee. But he drew these lovely pictures, and this is showing a wedge of the kidney. What we're looking at here is the site where the collecting duct empties into the renal calyx. If we follow this up, this is the collecting duct. The collecting duct is now receiving fluid from this distal convoluted tubule. If we go up here, this is the renal corpuscle. Let's follow this guy. Here's the renal corpuscle. This is Bowman's capsule, proximal convoluted tubule, the descending limb of the loop of the nephron, the hairpin turn, the ascending limb of the loop of the nephron, coming to the distal convoluted tubule, which now empties into the collecting duct, and the collecting duct now traverses the corticomedullary gradient to empty in the renal calyx. That's the structure of the nephrons. Now you'll notice that there are a whole bunch of other nephrons in here. The nephron that we're going to talk about today is this one. This one has a great long loop on it. That's called the juxtamedullary nephron. Only about 15% of the nephrons in the kidney are the juxtamedullary nephrons. But they have the job of setting up this gradient. And it's the gradient now that we're going to use to concentrate urine and to get rid of ions and molecules we don't want and keep the ones that we do. So what we're going to do is we're going to follow this structure, and we're going to follow it through the corticomedullary gradient. This next picture is one I like because it shows the details of everything we've talked about so far, but it gives them in terms of a single nephron. So again, up here, this is the afferent arteriole forming the glomerulus, nestled in Bowman's capsule. The afferent arteriole provides blood to the glomerular capillary, and the glomerular capillary is then drained by the efferent arteriole. The efferent arteriole now forms the structure we talked about last time, which is the peritubular capillary up here. This should be PERI, not para. This is the peritubular capillary up here. Then this is the Bowman's capsule emptying to the proximal convoluted tubule. This then enters into the descending limb of the loop of the nephron, makes the hairpin turn down here, and ascends as the ascending limb, and up here now enters the distal convoluted tubule, which is going to empty into the collecting duct. So that's the structure of the nephron. We're going to simplify this into a two-dimensional image and talk about what's going on at each level of this nephron as the descending and ascending limbs traverse the corticomedullary gradient. So any questions up to there? So what we've done so far is we've set up the gradient and we've placed this juxtamedullary nephron. Let me properly label this as the juxtamedullary nephron. And then over here on the right-hand side of the board, I want to begin talking about the epithelial cells that make up these structures. So we're going to label this as the epithelial cells of the descending limb. So we're going to draw a bunch of boxes that represent this region of the nephron. So here are the boxes. this is meant to be the descending limb. This is the lumen. So to orient you again, here's the basolateral surface. There's the basal lamina. And out here is interstitial space. That's just a fancy term for talking about the extracellular space around all these cells. So this is the extracellular space. This is cortical. This is the concentration of ions in the interstitial space is going to get higher and higher and higher. If we were to reflect what we've done over here, we could say that the concentration of ions up here is about 300 milliosmoles. But down here, down deep in the medulla, we're down around 1,200 in some cases, milliosmoles. So if you could imagine being inside this tube, if you were swimming down through this tube and you could look through the cells, you would see on the far side of the cells that it was getting saltier and saltier and saltier. Now, these epithelial cells of the descending limb are characterized by a set of transmembrane proteins known as aquaporins, AQP1. It's just a designation for there are about 8 of these, but this is the most common one. It's the only one, in fact, that's found in the descending limb. Now let's think about what this means. Aquaporins, of course, are water channels. They're in both the apical and basilar membranes. So now water can leave the lumen, flow into the cells, flow through the cells, and flow out the other side. As the water leaves the lumen of the descending limb, it comes to equilibrium. The fluid inside the lumen comes to equilibrium with what's on the outside. So the concentrations we would read out here, pick a number, let's say this is 400. We get pretty close to 400 milliosmoles on the inside. It's come to equilibrium. But it achieves that equilibrium only by allowing water to leave. One of the hallmarks of the descending limb is that it lacks ion channels. So the ions are trapped in here. They can't leave. Water can leave, but the ions can't. So as the water leaves, the concentration of ions gets higher and higher in here. And as the water leaves, the concentration of ions in here comes to equilibrium with whatever it is in the outside world. So it tells us that as the fluid is moving through the descending limb, the concentration of ions inside the lumen of the tube is equilibrium with what's ever on the outside. So if this is 600 inside, is 750 inside. And we reach that equilibrium simply by allowing water to exit. Then we come around the hairpin turn. We come around the hairpin turn. Now we're going to convert these epithelial cells to a different type. These are going to still be epithelial cells. We're going to change the proteins in them. So we come around the hairpin turn. I always have trouble with this for reasons I don't understand. Oh, quite good. So this is the descending limb. Over here, this is going to be the ascending limb. So the fluid flow here is from the descending limb around the hairpin turn, and now enters the ascending limb. So this is equivalent to what we see here. And the fluid flow is from the descending limb around the hairpin turn and into the ascending limb. All along the descending limb, the concentration ions inside the equilibrium with what's on the outside. After we come around the curve, we change the kinds of proteins we have in these epithelial cells. So that when we come around the hairpin turn, these cells lack aquaporins. So they come around the hairpin turn, and now there are no aquaporins to let the water out. But instead, what we replace them with are ion channels. So they lack aquaporin, but contain ion channels. And it's through these ion channels that the ions can now move down their concentration gradient. And as the interstitial fluid concentrations get less and less and less as we go up toward the cortex, we come to equilibrium. This region, right after the hairpin turn, this region in here has a name. This is said to be the this is said to be the thin segment. The thin segment is characterized by ion channels. Then as we come around the hairpin turn, as the equilibrium is reached at each of these points, as we get higher and higher in the corticomedullary gradient, so we get closer and closer to the cortex, we now enter a region of the ascending limb, which has another name, and this is called the thick segment. And what characterizes the thick segment here is that the apical surfaces contain ion transporters. The ion transporter that we find in the thick segment, the ion transporter in the thick segment, we're going to enter the thick segment up here, is something called the sodium potassium transporter. So I'll draw a couple more examples of epithelial cells here, which are meant to be thick segment. I'm sorry, meant to be thin segment. And now we enter here. Make these a little longer so we get a sense that this is the thick segment. So these are epithelial cells of the thick segment. If we look carefully at these cells, and again, remember, this is luminal. This is lumen here. On the apical side, or on the luminal side of these epithelial cells, we now begin to see a new set of proteins. And these new proteins are the sodium potassium 2 chloride transporters. So what these proteins are doing is they're snagging sodium ions and potassium ions and chloride ions that are in the lumen. And they're moving those ions into the epithelial cells. And then on the basilar surfaces of these epithelial cells is going to be the sodium potassium ATPase. because these transporters are pretty efficient, they're going to fill these cells with sodium, potassium, and chloride. They're like little buckets, and so they're going to get filled. To keep them from filling up, we now pump the sodium out the basolateral surface. Turns out there's always so much potassium inside these cells, all you have to do is put some basilar channels in, the potassium is going to run out. Chloride will follow the sodium. So in this way now, we are going to be moving the ions across the cell. And initially, we're simply going to come to equilibrium. So we're coming to equilibrium, but as we approach the distal convoluted, as the fluid in the tube gets, then these transporters are so efficient that the concentration of ions in the lumen of the ascending limb falls to about 50 millimolar sodium and 50 millimolar chloride. So where at first we're just reaching equilibrium, as we get higher and higher here, we reach a point where we have about 50 millimolar sodium and 50 millimolar chloride. Well, if you add those up, that's 100 milliosmoles. We said that the osmolarity of the fluid in the 300 are actually hypoosmotic once this fluid leaves this ascending limb and enters the distal convoluted tubule. Now, the distal convoluted tubule is going to be the final place where we can reclaim sodium. It's a bit of a fib. Again, as I said in the introduction, the distinction of the cells between the distal convoluted tubule and the initial portions of the collecting duct get a little bit hazy. But regardless, we can think about this as we're going to say the distal convoluted tubule is the site where we're going to last job, it's the last station where we can remove the sodium and chloride. And that's where we're headed next. So up to now, we have used either the aquaporins or the ion channels or the transporters to bring things to equilibrium between the lumen of the tube and the extracellular space. But as we approach the distal convoluted tubule, the transporters have been so efficient that we can actually approach a level of hypoosmolality. But we're going to remove this as well in the distal convoluted tubule. So any questions to there? The next stop is going to be the distal convoluted tubule. So let's use our boxes again. And up here, draw a series of boxes that are going to represent the distal convoluted tubule. So this is the lumen of the distal convoluted tubule. In the lumen of the distal convoluted tubule, we have about 50 millimolar sodium and about 50 millimolar chloride ions. So it's in the neighborhood of 100 milliosmoles. Now, this is the last place where we can reclaim or reabsorb sodium. So the way we're going to do this is out on the apical surfaces of these epithelial cells. Will be a transmembrane protein, which is said to be an epithelial sodium channel. You'll often see this referred to as an ENaC, E-N-C, epithelial sodium channel. So the ENaCs are out on the apical surfaces. Remember the concentration of sodium. We never get concentrations outside the range of about 10 millimolar sodium in the cytoplasm. What this is telling us is that we can simply use the gradient. We have about 5 times as much sodium in the outside as the inside, so we can simply allow the sodium to move down its concentration gradient. But again, these little buckets are going to fill with sodium fairly fast. So we need a way of clearing the sodium. So on the basolateral surfaces, and again, we put in a basal lamina here. On the basolateral surfaces of these epithelial cells, it's going to be another protein. This is going to be, again, a sodium potassium ATPase. So it's busily burning ATP to pump the sodium outside the cell. Just as we saw in the proximal convoluted tubule, there's going to be a peritubular capillary that wends its way around the tubule. This is said to be peritubular capillary. This peritubular capillary is now going to feed into the venous system. It's going to drain the kidney, but this is the last place where we can pick up sodium. Now, in our this isn't so much of a problem. In our modern world, we have so much sodium in our diet that it's actually probably kind of dangerous to us in how much sodium we eat. We have way more sodium than we need. But historically, you don't have to go back very far in time, and there just simply wasn't that much sodium in people's diets. And so there is this biological pressure now to try to conserve sodium. And this is pretty much the last place where we can do the conservation of sodium. And the reabsorption of sodium in the distal convoluted tubule is under hormonal control. Specifically, it's under the control of aldosterone. Sterone, that tells you it's a steroid hormone. It's a steroid hormone It's a steroid hormone secreted from the adrenal cortex. It's a relative of cortisol, but it's now being used here in these epithelial cells. And let's look to see how it works. If this happens to be an epithelial cell in the distal convoluted tubule, that epithelial cell in the distal convoluted tubule will contain receptors. These will be aldosterone receptors. When the aldosterone comes in, it binds to that receptor. And you'll remember the sequence of events of steroid hormones. The aldosterone binds to the steroid hormone receptor, and that will then lead that receptor hormone complex to the nucleus. In the nucleus, this aldosterone receptor complex generates transcription factor. In this particular cell, in this particular case, with this particular receptor, this transcription factor works on genes in the nucleus to upregulate the production of sodium channels, the ENACs, and the sodium-potassium ATPases. So under circumstances where you don't have very much sodium in your diet, And there are signals coming from your hypothalamus that says, there's not enough sodium around here. See if you can scavenge up some more. What that does is it signals the cortex to release aldosterone. The aldosterone binds to the cytoplasmic receptors in these epithelial cells, upregulates the number of sodium channels so that now we're going to conserve more sodium. We're going to bring the sodium into the cell, pump it out through the sodium-potassium ATPase, and put it back in the blood. Now, in our modern world, what this often leads to is hypertension. Not in everyone, but some people tend not to deal with salt as well as others. And so when they take excess salt in, they'll actually simply pass it through the enax, put it back in the blood, and water will follow. Water always follows the sodium. When the water follows the sodium, it fills up this closed compartment, and blood pressure goes up. So one of the ways we deal with hypertension is we give people drugs to affect these sodium channels. Basically block the sodium channel and let less sodium come in. We reclaim less sodium. It spills over into the collecting duct and goes out to the outside world. And now we reduce the amount of sodium moving into the blood vascular compartment, and in this way we can lower blood pressures. Many of these drugs also contain another drug which blocks something called the sodium chloride cotransporter, but it's the same effect. Reduce the amount of sodium going back into the blood vascular compartment, which will thereby increase the volume of the blood vascular compartment because water follows. So we try to avoid that and let this excess sodium spill out into the collecting duct and go to the outside world. But that's pretty much a modern problem because we don't have so much. Historically, we didn't have so much sodium in our diets, but now with so much prepared food, almost everything we eat has more sodium than we ever possibly could need. Yes?
So what is the role of the sodium potassium?
What's the role of the sodium potassium? Okay. I'm sorry?
What's the potassium doing?
What's the potassium doing? The potassium, in a way, the potassium can't help itself. The way we use these pumps is they're coupled. 3 sodium out, 2 potassium in. So in this case, we're pumping the sodium out of the cell. But you're right, we're going to fill these vessels up with potassium. But we don't really have to have much of a problem with that. We simply have to have potassium leak channels in the basolateral membrane. And because we have 110 millimolar potassium inside the cell and 4 to 5 on the outside, it's going to flow out. And it will return to the blood in that way. So we talk about the sodium-potassium pumps, but they're mostly functioning to get sodium out of the cell. But you're right, they're going to bring potassium in. For every 3 sodiums we pump out, we're going to bring 2 potassiums in. Now, if you didn't have some exit strategy for the potassium, then the potassium concentrations inside the cell will go up and up and up. But all you need to do is have a few channels, a few potassium channels, and it runs down concentration gradient because there's always 110 millimolar potassium on the inside, 4 to 5 on the outside. It's a Whomping gradient. Just let the potassium pour back out again. And in this neighborhood, because it's surrounded by the peritubular capillary, it can go back into the blood. Shall I tell you the story? Yeah, in the last minute, I'm running out of time anyway. You're right about potassium. Potassium is this ion that I don't like to talk about much in here because we have excess potassium in our diet. Everything we eat, whether it's plant-based or animal-based, the inside of that cell has loads of potassium in it. So every day we get an excess of potassium in our diet. And there are going to be cells in the next step. We're not going to talk much about it, but once we get over here into the collecting duct, there are going to be special principal cells. And these principal cells are busily taking any extra potassium that's in the extracellular space and moving it into the lumen of the collecting duct so it can go. And those principal cells are also under hormonal control. They're also under aldosterone control. And so they're getting rid of excess potassium. And as I mentioned early on, if you let your potassium concentrations in the plasma get much above 4 to 5 millimolar, then you're fouling the nerve cells and the muscle cells because you're depolarizing the membrane potential of those cells. I've asked that question a number of times on exams, trying to drive home the idea of how elevated extracellular potassium will depolarize cells. And that depolarization, if you remember back to the pacemaker cells, will stop them from functioning. I'll leave it with that, and we'll come back to the story and get things into the collecting duct on Monday.